Okay, as time is precious and we had some IT issues, I'm going to start this talk and just begin by saying my deepest thanks to the organizers for letting me speak, for accepting the talk. And also, of course, I wanted to tell all of you that for every moment I am here, I celebrate your success. Thank you. We are endeavoring today to explore many facets of the results of many decades of investment into the concept of high-dimensional data and bioinformatics. And I celebrated that here in this country and across the continent simply because I believe that it is a step into the future that is relatively low cost. Kader Asmal, the Minister of Education in this country at some time, came to me one day and said, I really like you, because all you cost us is a bit more than a philosopher. For them, it's a pencil and some paper, but for you, it's a computer. We'll get by. In any case, what I want to talk about today is precision medicine, but this needs to be first acknowledged that one of our directors at SAMBI has passed, and those of you that were influenced by him, please reflect. We have a significant problem in modern biomedical research, and that is the problem of finding appropriate drug targets. You heard a fascinating presentation in the beginning of this whole meeting about the difficulty of selecting targets that don't give you off-target effects. And in fact, the strategy today, as we've just heard a few moments ago, is to look at the genome to see where is code that is influencing the disease. This results in what I call gene choice, and that means that somebody somewhere wants to look at a gene. The problem with that concept is that genes are easy to find. They're relatively easy to find. You can find genes associated with pretty much anything. Balding is one of them I'm particularly interested in. And the issue around this is that once you've discovered a gene associated with a disease, you become passionate about it. There becomes bias. And within biotechnology and biomedicine, I have seen this. I've sat inside a biotechnology company investing 200 million in a target. And it was a target day. Just as we had clickers the other day with ABI here, now we clicked and said, I'm choosing this gene, that gene. That was a $200 million investment at the end of the day. What is the success rate for Alzheimer's disease drug discovery? Besides partial remission of cognitive loss in early stages of Alzheimer's through addressing A beta pathology through antibody treatment, there is no way to address Alzheimer's. And of all of the gene-based trials, none have succeeded, and there have been thousands of them. So the issue is that there needs to be a new paradigm. So the paradigm we're following in my laboratory and have for some time, for those of you who know me, is a relatively systems-approached methodology where we don't go after genes, but then the issue becomes how do you drug a system? So we've worked on that problem, and over several years I've looked at this from the perspective of condensing information from the transcriptome, which is not your friend, into pathways which we know, but they're not our friends either, but at least we know them. It's that kind of situation. Pathways are a human construct. They don't exist, but we use them as functional units, and that's enough. So this is work I've done with Purya Naderi, who's an instructor in my group. And Purya has been fascinated by the problem of how do you get from the cells in the dish to a successful human trial. And we've written a paper about this problem, and it's in Neuron. If those of you want to look at the underlying hardcore bioinformatics, you can find it. And what I want to stress in this problem is that we're really looking to try and make success in models bridge the gap to make success in human patients. And that gap is where we're trying to hypothesize, which ways can we improve that? One of the things that we assume is that there has to be a failure. Can it be in the drug, in the choice of model, or in the target? Those are the three that should represent the basic concepts. And the issue there is that from a single gene that you might have in one of your models in the mouse or in the dish that you're disrupting to cause the phenotype, you don't really know what the relationship is between that disrupted mutant phenotype and the disease, which you see as a phenotype of that model. The intervening complex cascade is pretty much, black box. So you have to investigate that, and that's the process of science. But then is it a true reflection of the human condition. So that's the problem we're trying to address. So 6 million people in the United States alone afflicted with Alzheimer's. All of us in this room are probably creating Alzheimer's pathology as I speak. And I still have trouble remembering names. And the point being that it is an insidious disease that we all suffer from. Some of us die from it. Some of us don't have it by the time, don't die from it by the time we go there. Models represent sporadic or genetic or specific single gene. And the issue is that most of the models that are used for Alzheimer's are driven by this single gene concept. And we don't really know how sporadic Alzheimer's is caused. And yet that is the major part of the disease. So we'd really like to get under this problem of how well a model disease actually mechanism matches the human disease. And if we can solve part of that, that's progress. And that's really what we've been trying to do. If you think about a pathogenic cascade, and I've been hinting about the concept of that, it's a set of genes in a pathway, and transcription factors and gene products and signaling pathways. Different ones are going to be active in the model to the human. They may have the same pathway, but different elements are going to contribute. So you need to understand how to equivalentize that pathogenic cascade. So one of the things that we use is pathways, and because of those, we can reduce the noise, we can use them as a unit of activity, and if we take a pathway activity, it can be positive or negative. So we're not thinking like gene set enrichment here, we're thinking activity of genes in a fixed pathway, positive or negative. Is there some form of way we can look at that? I'll show you how. We're calling this IPAA. But the take home here is we can get directionality. The pathway can be up or down. From that, we can then say, we can choose the pathways which are all up in the model or down in the model and match them with those in the brain and vice versa. So that means we have portable function. Okay? And it means there's no background. We don't have an issue about the enrichment background or the size of the pathway. We can do some cutoffs on that, but we can just look at each pathway individually. So if we do that, I'll take you through a sort of schematic of what we do. On the genes on the left, samples across the top, disease control. So this is a sort of sample matrix. And if you take any of the pathways or all of the pathways from a popular resource like MSigDB, you can start to perform pathway activity analysis. And this is a long legacy of research that's been done in my group. So anybody interested, let me know. Take home from this is that you can use each pathway. So let's just say this is the Wnt pathway as a mask. And here I show you 4 elements of the Wnt pathway. But if you rank all of the gene expression in the control and all of the gene expression in the case, you don't do a differential gene expression analysis. You just take the rank and then you say, what is the number of the rank of my gene in the sample on the left? And then you take a look and you just rank it according to pathway rank. So, 7, 79, 112. And then the representation of that you can map out between disease control, and you can do your biostatistics to ensure that you have a representation that is basically a form of mean. And you can take the mean rank of the activity of that pathway. Now, I've had lots and lots and lots of papers on and discussions and issues around the algorithmic choices here. Happy to defend them. They're pretty much dynamic. We keep changing them. The bottom line is we keep trying to improve this metric. The take home is that you can design then experiments that are very comparative, where you don't have to do differential gene expression, but instead you have a set of summed activities. That allows you then to look at human genes in the human brain, different regions of the brain if you choose, and then take a look at the known pathways and find a common set of pathways that show similar up and down dysregulation in the brain across models, across brain regions. You can do the same for any human-derived or mouse or other model and take a look at the known pathways. But here then, you can match the dysregulation that I see in the Venn diagram with the dysregulation in the model. And I'll show you the data that represents that. And that's the hypothesis that we used. We didn't know if it worked. So what comes from that then, first of all, are core dysregulated pathways that you're quite confident are showing the same behavior across different subjects and different areas of the human brain, if that's what you're looking for. And then we can go and look at those in the models and see if we can drug them. And that's basically what we did. So the experiment is made up of a cohorts, from which we could find different gene expression products from different regions of the brain. And we looked here at the temporal cortex and parahippocampal gyrus, and we found an overlap of, as you can read, 277 pathways, which are in common between these two systems. We added another system in another experiment, but you can see that if you do heat maps It looks similar, which you can eyeball here, but what we were able to find then is common sets of pathways which were common between these two stories. What about gene expression? Can't you just do this with gene expression? It's just going to be simpler, right? The problem with gene expression is variance. You get a lot of variability. And as you know, high-dimensional data and variability together are not good friends. And what comes with that is a plot on the right. So the regression gives you about 0.51 or about 0.84. And obviously, 0.84 is a better number, so we go with pathways. This is a schematic of the models. I do not expect you to dive hard into this, but what I do want to point out is that on the left, we have a set of mutations, lesions that generate the pathology. The pathology is A-beta pathology, so you've got sheets of dysregulated protein, and that then induces in the model tau pathology, which are neurofibrillary tangles which appear within the cells of within the neurons. If you have a version of A-beta called 40 or a version of A-beta 42, and that has a particular ratio, that is either a good ratio or a bad ratio. Think about lipids and lipids and things like that. And the good ratio is good, it's protective. The bad ratio is where you get Alzheimer's. So this is how the model works. And we discovered that if we took a look at these models in more detail, and I don't have a pointer, but if you take a look at the top, you see it says A-beta 42 high versus A-beta 40. That is the model that turned out to be clustering on a heat map with human. And the rest of the models did not, cellular models. This is weird, because as you can imagine, cellular models are not likely to be very human-like. So if we cluster based upon these pathways and the correlation of these pathways, we discover there's a 30% correlation between the human and the model. And that's quite high, because if you think about it, it's in a dish. So that was a point of contention. If the reviewers in the audience, thank you for that question. We answered it, right? Okay, so it's got published. So how am I doing for time? Okay. What I wanted to make here is that if you take a look at the expression of our top pathways in the different systems across here, you can see that the significant difference at the gene level is very different with respect to how we see differences at the pathway level. And if you take a look at the interactions of the pathways, which is this plot on the right, if you can plot their correlations, you find that groups of them come under common functions, but do not share overlaps. So if we go in now and test this, on the left-hand side is a blot, and you can see phosphorylated tau at the top right appearing, not in the control, but in the model, okay? And then you can see it's measurable. If you then measure the effect in a mouse, you can see the same sort of thing. If you then measure the effect of adding an inhibitor, you can reduce that p-tau. And where I want to go with this is clinical trials. So for the last couple of minutes, we tested 3 drugs. The first was losmapimod, and that did what we wanted it to do. That actually is a drug which has been trialed, so it's a repurpose for a cancer treatment. And it's very low toxicity. And actually, when we treated models with this, they did pretty well. They lost neuroinflammation, kept cognitive strength, etc. So it's been cool. Dorapimod did not work. It was toxic. And then we took another one, the optimistically named VX745. What's intriguing about this third one is that one is the one we actually tested first, because that one was in trial for Alzheimer's-related pathology. And that trial failed. So think about this. What we were able to do is show that it had no selectivity in our system and did not work. We were able to do that in the middle of that trial. I wish I'd had stock in the company, but the point is we were able to predict the outcome of the trial. So that's probably how helped us get published. But the point I'm trying to make is that this is a very powerful technique, because it can at least tell you where not to go. Reduction in risk is one of the key facets of bioinformatics. And here you can see it in play. So this drives translation. That's the point. And we think pathways represent pretty powerful targets, and they allow us to make portable function. And this tool, we think, is an accelerator for drug discovery, because soup to nuts, despite the publication, we were actually able to do this kind of work in a period of 4 to 6 weeks. If you think about that, that's a nice way to start developing a drug system. So we're open for business. Anybody wants to cure Alzheimer's, come see me. But seriously, I think this is a powerful way to go to the future. Final point I want to make is that you can step down through the pathway, and you can start to look at individual impacts and get finer and finer resolution drugging. This way, you're quite confident about the pathway you've chosen. What you're not confident about is, of course, the interactions and the off-target effects. Thank you for your attention.
much, Winston. So we have time for one question.
Thank you for a very informative talk. My question is that if we have the data set that has the samples and the genes, and the metadata that has either the control and the disease, the genes are the same, right? For both the control and the disease.
The potential expression of the genes is the same,
Yes.
correct?
Then it means that the gene sets that you're going to use for the control and the disease for the pathways are the same, which means you're going to get the same pathways, the activation in the same pathways. Now, how do you identify the ones that are upregulated and the ones that are downregulated as for the pathways?
Well, you don't actually know whether they're up or down regulated, but what you do know is that the same genes are present in your case and control. So let's say you're looking at your case and you see that there's a net increase in expression of that pathway. You can measure that using a blot or something. You want to find out if your bioinformatics supports that. What you do is you take a look at the expression level of each of the genes that are in that pathway. So you look at the first gene in that pathway and you ask, what is its expression level in the corpus that you're measuring? Oh, it's one of the most highly expressed genes. So its rank is 1 or 2 or 3. Taking those ranks and taking their average gives you a positive or negative value. And if it's a positive value, the pathway is active. Okay? That's as simple as that. I mean, there's stats, but that's it. Okay.